Hello everyone and welcome to the biology lecture series. Okay, and today we'll be talking about the chapter heredity and inheritance. So in the previous lectures we were studied about the heredity as well as the inheritance, how can we define it. Then we studied about the variations and what are the causes of these variations. Then we studied about the Mendel's experiment, okay, where we studied about some of the terms related to the genetics as well as the inheritance. Then we also talked about the monohybrid, trihybrid and the dihybrid cross. Clear? And then we also talked about some of the terms such as test cross as well as the back cross. So in the previous session, we were talking about the monohybrid cross and we talked about the phenotypic and the genotypic ratio which is observed in the monohybrid cross. Clear? So this lecture is the continuation of the previous lecture. And in this lecture, again, we'll talk about the Mendel's law of inheritance. Okay, so what were the laws that the Mendel gave? These are also known as the Mendel laws. So as you know, the Mendel, he conducted experiment on the garden pea, and based on his experiments, okay, he concluded these four laws. Clear? Or he formulated these four laws. So let's start with this lecture now. So on the basis of the experiment performed and the results obtained, Mendel formulated four laws. So first law is the principle of the paired factors. Second is the principle of the dominance, also known as the law of dominance. Then next is the principle of the segregation, also known as the law of segregation. And then the fourth one is the principle of independent assortment, also known as the law of independent assortment. Now we'll study each of this law, okay, in detail. First, let's talk about the principle of the paired factors. Now each character in an individual is governed by two factors known as the genes. So Mendel, what he concluded that there are some factors that are responsible for the inheritance. Clear? So he didn't give the term gene. So he said there are some factors. Clear? Later on, these factors were found out to be the genes. Clear? And these factors, okay, they are the basis of the inheritance. Or you can say they lead to the inheritance. Okay? Clear? Or they are the basis of the inheritance. Clear? Okay, so this is known as the principle of paired factors. So there are two factors, okay, which govern any character, and these are now known as the gene. Now the alternative form of the gene is called as the allele or the allelomorph. Okay, what does this mean? This means that if we talk about the character height, okay, so there are two alleles for the height. One is the dominant allele. So this one is the dominant. Clear? And then we have the small t. This one is the recessive. Okay, one is dominant allele, this one is the recessive allele. So there are two alleles, and any gene, it has two alleles. Okay, and the alternative form of the gene, these are known as alleles. Clear? If we talk about the, you can say, shape of the seed. Okay, shape of the seed. So this is another character or the trait. Then suppose R is for the round, okay, and small r is for the wrinkled. So again, this gene for the shape of the seed, it has two alleles. One is the capital R, another one is the small r. One is dominant. Suppose capital R is the dominant, so the round shape is dominant, and the other one is recessive. Clear? Now, if an individual consists of similar type of alleles, okay, they are known as the homozygous. Means capital T or small t, small t. So these are the homozygous individual. Capital T, capital will be homozygous dominant, small t will be homozygous recessive. So capital T and capital T, this is homozygous dominant, and small t, this is homozygous recessive. Clear? But if the organisms, they have different type of alleles, these are known as heterozygous. So heterozygous will be capital T and small t. So this is the heterozygous condition. Clear? Now, let's talk about this. The principle of dominance, also known as the law of dominance. So, when two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrids are dominant character, and those that do not appear in the F1 are the recessive characters. So this is known as the law of dominance, where the dominant character or the character which appears in the F1 is known as the dominant character, and the character which is not expressed is known as the recessive character. So again, we can see this by the example. So if the tall individual, it is crossed with the dwarf individual, then what will be the F1 produced here, or the hybrid produced? Capital T and small t. And this will be the tall. What will be the phenotype? It will be the tall phenotype. Then here you can see the dominant. So capital T is the dominant gene here. Okay, so this one is the dominant. So that's why the phenotype is tall. So the character which appears in the F1 generation, so this is the F1 hybrid, is known as the dominant character, and this allele is known as the dominant allele. And you can see small t is recessive. Okay, this is the recessive allele. That's why its expression is masked by the dominant allele. So you can write here expression of recessive allele, or you can say a recessive gene, is masked, okay, by the dominant allele, or you can say the gene. Clear? So this is known as the law of dominance. Now let's talk about the law of segregation, also known as the principle of the segregation. Now, according to the law of segregation, or this law states that when a pair of the contrasting factors, also known as the genes or alleles, they are brought together in a heterozygous condition. If they are brought together in the heterozygous condition, the two remains together without being contaminated. But when the gametes are formed from them, the two separate out from each other. Clear? This is also known as Mendel's first law of heredity. So law of segregation is the Mendel's first law of heredity. It is also known as law of purity of gametes. Clear? So what does this mean? This means that the law of segregation of the gametes, which are proposed by the Mendel, simply state that during the formation of gametes, means during the formation of sperm and the egg cell. So both of these are the gametes. Sperm is the male gamete, egg cell is the female gamete. The two alleles for each gene, it segregates from each other, alright, so that each gamete carries one allele for each gene. Again, we can understand with this example. So if we have, okay, T and small t, okay, then what happens? One of the T, so what are the gametes formed here? Okay, gametes formed will be T and the small t. So you can see the two alleles for each gene. Okay, the two alleles here, capital T and small t, They segregate during the gamete formation. Clear? So this allele will form, or you can say this capital T, okay, will go in one gamete. This small t will go in another gamete. Clear? This is clear. So alleles, okay, they segregate from each other. This is known as the law of the segregation, and each gamete will receive each allele. Okay. Another case you can take where capital T. So here gametes form will be T. Okay, all the gametes will be capital T only. Here, but here it is clearly visible how the segregation of the alleles is taking place into each gamete. So one allele goes in one gamete. This is clear. This is known as law of segregation. Now let's talk about the law of independent assortment, also known as the principle of independent assortment. Now, if the inheritance of more than one pair of characters, okay, is studied simultaneously. More than one characters means if we are talking about the dihybrid cross, suppose. Okay, so in dihybrid cross, we study the inheritance pattern of two pair of characters. Okay, two pair of contrasting characters. Clear? So like you can take the yellow seeds which are round or green seeds which are wrinkled. So there are two pairs of character. Clear? So one pair can be, you can say, the color of the flower or the flower color, and second can be the shape of the seed. Clear? So if we are talking about the dihybrid cross, earlier we were talking about the monohybrid cross, but if we have to study the law of independent assortment, we will talk about the dihybrid cross. So what happens? If the inheritance of more than one pair of character is studied simultaneously, the factors or the genes for each pair of characters assort out independently. Now it is called as the Mendel's second law of heredity as well. Here what happens? The assortment of alleles for one gene is independent of the assortment of allele for the another gene. Clear? Again, we'll read it. The assortment of alleles of one gene. So, okay. So, one gene, suppose we take the gene for the flower color. Okay. So, this is one trait that is controlled by one gene. Okay. So, here, we take another gene or the another character. Suppose we take the shape of the seed. Clear? So assortment of allele of one gene. So flower color, okay, is suppose controlled by the gene Y. Capital Y is for the yellow color, and then we have the small y. Okay, whereas shape of the seed, if we talk about, it can be round or it can be wrinkled. So it is controlled by R, which gives you the round seed, and you can say small r, which give you the wrinkled seed. Clear? So these alleles, you can see, these will assort out independently of these two. Clear? This is clear? So the alleles of one gene, so these alleles will assort independently, okay, from these genes. Clear? And this is seen in the dihybrid cross. Clear? This is known as the law of independent assortment, and this is also the Mendel's second law of heredity. Clear? Now let's move on. So if in a dihybrid cross, the Mendel crossed genetically pure yellow round seeds. Okay, so here we have two pair of contrasting characters, yellow, okay, and round seed. They were crossed by green and the wrinkled pea plant. So what will happen? All the plants of the F1 were yellow as well as they had the round seed. So this was the genotype of the F1 hybrid. Clear? Now Again, you can see this was the cross that was initially made. So these were the parents. Clear? Here, what will be the gametes produced? Y and capital R and Y and capital R. Clear? So Y and capital R as well as Y and capital R. So these are the gametes which are produced. Alright, again here, small y and small r and small y and small r. So these are the gametes which are produced by this parent. Now, if we make a cross here, then you can say, you will see what is the genotype of the F1. All have the same genotype. So F1 plants are all yellow and round seeded. Clear? Now, let's talk about the F2 generation. Alright, if we self the F1, after selfing of the F1, we get the F2 generation. But here 4 types of plants are produced. Okay, yellow and rounded, yellow and wrinkled, green and round, and green and wrinkled. So this is the phenotypic ratio that we get in the F2 generation. Clear? And F2 generation is obtained by what? Selfing of the F1. By selfing of the F1, you will get the F2 generation, and the ratio or the phenotypic ratio is 9:3:3:1. 9 are yellow rounded, 3 are yellow wrinkled, 3 are green and round, and 1 is green wrinkled. So here you can see the alleles of the one gene, they assort independently from the allele of the other gene. Clear? So this is the method to show you or to explain you the law of independent assortment. Clear? So again, here it is showing the selfing of the F1. So this was the F1 that was produced. It is self now. And these are the gametes that are produced by the parents. So what are the gametes? Capital Y, capital R, capital Y and small r, small y and capital R, and small y and small r. Again here, same gametes are produced. So you can see here, the alleles of one gene is assorting independently, alright, from the alleles of the other gene. Clear? Because you can see here the gametes. You can see yellow and red. Then yellow, and this one is the wrinkled. Then again green. Green is now pairing with the round one. Clear? So this shows you the what? Law of independent assortment. Now, if you make a Punnett square, and from the Punnett square you can find out the genotypes of the F2 progeny. So here you can see Y and Y, capital Y, capital Y, capital R, capital R. Okay, so these are all the phenotypes that you get. Clear? Now the ratio was 9:3:3:1. So let's first talk about yellow rounded seed. Okay, yellow rounded seed. Okay, means both are dominant. Okay, both are dominant traits. So here this is the first one. Okay, this is the second one. Then this is the third one. This should be the fourth. This is also yellow rounded. This is fifth, again yellow rounded. This is sixth, yellow and rounded. Then seventh. This is also yellow and rounded. This is also yellow and rounded. So eighth. And then which one with the yellow and rounded? This one also. This is the ninth one. Okay. And what was the ratio? 9. Okay, they appear in the ratio of 9. Then we have yellow and wrinkled. Okay, yellow and wrinkled. So these were 3. So yellow is comes from the dominant gene. Clear? So here, dominant one should be dominant and another one should be smaller and smaller. Then only we will get the wrinkled condition. So let's look for smaller and smaller. So this should be the first one, yellow and wrinkled. Then again, this should be yellow and wrinkled. So this is the second one. Then again, smaller and smaller is where? Here again. So this is the third one. This should also be yellow and wrinkled. Then we have green and round. Okay. Green and round. This should be again 3. So for green, this should be, Y should be non-capital. Okay, it should be in small letters. Clear? And round means one of the genes should be dominant. Clear? So what is the green here? We'll find the small y now. Clear? So where is small y and small y? Here. Okay, small y, and so this is green and round. So this is the first one. This is also the second, green and round. And then this is also green and round. So 3. Now the last one is green and wrinkled. Okay. Green and wrinkled. Which is 1. Means this one. Alright, both the genes should be, or the alleles, all the alleles should be in the recessive condition. Then you will get the small y, small r. Clear? So we know that the capital Y, okay, the capital Y, it is for yellow. Small y was for green. Capital R was for round. And small r is for wrinkled. Clear? So this is the F2 generation of a dihybrid cross. And the phenotypic ratio is here 9:3:3:1. This is clear? So by this method, you can show the law of independent assortment. The alleles of the one gene, okay? So these are the 2 alleles of one gene. They assort independently from the alleles of the another gene. Clear? Okay. Now let's talk about the genes. What are genes? We were talking about genes for such a long time. Now let me introduce you to the term genes. Now the term gene was introduced by Johansen. So Johansen was the person who introduced these terms for the Mendelian factors. So Mendel, okay, he talked about the factors. There was some factors that was responsible for the inheritance. But later on, these factors was found out to be the gene. Clear? Now, the gene determines the physical as well as the physiological characteristic. Clear? Both physical as well as the physiological characteristic. So it is responsible for, you can say, the morphology or the phenotype also, and it is also responsible for the physiological characteristic as well. Now they are transmitted from parents to their offsprings, okay? Generation after generation, means they are inherited. Okay? They are inherited from the parents to the offsprings. Clear? This is known as inheritance. Or you can say the transmitters of genes from the parent to the offsprings, from generation to generation. This is known as inheritance. Now, where are the genes located? So these genes are located on the chromosomes, where they occupy specific position called the locus. So what is a locus? So locus is the place where the genes are located. Clear? So it is a specific position where the genes are located on the chromosomes. So this is known as the locus. Clear? Now this was proved experimentally by Boveri and the Sutton. Okay, so this is known as the Sutton and Boveri, and it was proved that the genes are present at a particular position that is known as the locus. So this was done by Boveri and Sutton in 1902. Now these genes, they are responsible for the characteristic feature of an organism. Okay, so genes, they code for a specific protein, you know. Okay, genes are made up of DNA. Clear? And this DNA code for a particular protein, and this protein gives the particular phenotype to the individual. So you can see there are a lot of genes present in the human. Okay, so there is a gene for, if in plants as well. So if we talk about gene for the height, Okay, so they will give you, they will be responsible for tall height or small height. Clear? So there are genes for other features also, like skin color as well. So these genes are responsible for the characteristic feature of the organism, whether it is tall, whether it is short. Okay, if we talk about plant, we can talk about the flower color, whether the flower color is white or whether it is red. Then if we talk about the seed shape, whether the seed shape is round or it is wrinkled. We can talk about the position of the flower, whether it is axial or terminal. Clear? So all of these are determined by the genes. Now let's talk about the molecular structure of the gene. Clear? So genes, they are chemically, genes are DNA. They are made up of DNA, which is deoxyribose nucleic acid. Clear? This is clear? So genes, they are made up of the DNA, and you know DNA is transcribed into RNA, and RNA is translated into proteins. Clear? This is known as the central dogma. So chemically, the gene is a DNA. Now it consists of the following parts. What are the parts of the genes? First is the recon. What is the recon? So it is the smallest unit of the DNA capable of undergoing crossing over as well as the recombination. This is known as a recon. Smallest unit of the DNA that is capable of undergoing crossing over and the recombination. And because of this crossing over and recombination, what is produced? So this is the source of the variation. Okay. So because of this crossing over and recombination, there is the exchange of the genetic material. Or you can say there is exchange of DNA. DNA is the genetic material. Clear? Now this was the recon. Now another term is muton. What is muton? So it is the smallest unit of DNA which is capable of undergoing mutation. Clear? So mutation can be single base pair mutation. They can be changed in the single base pair. Okay, this is known as single nucleotide polymorphism. So muton is the smallest unit of the DNA that can undergo mutation. Clear? So mutation can be by a change in the base pair. Clear? For example, if you have the AUG sequence, okay, the sequence can change into, you can say A, or instead of uracil, you will get some another nitrogen base, or instead of this G, you will get another. So it can be AU. Instead of G, you get what? You get A. Okay. Now next is the cistron. What is the cistron? So it is the gene in the real sense, okay, which consists of number of the nucleotides and which is capable of synthesizing a polypeptide chain of the enzymes. This is known as the cistron. So cistron is basically the coding region. All right. And it is a gene in real sense, and it consists of lot of nucleotide, means it consists of a polynucleotide. So it is a polynucleotide, you can say. It consists of number of nucleotide, means it is a polynucleotide. Clear? And it is capable of synthesizing a polypeptide chain of enzymes. So it will code for a particular enzyme or the protein. So you can say cistron, it will code for a particular polypeptide chain or a protein. Clear? Now what is replicon? It is the unit of the replication. So what is replication? It is involved in the synthesis of the DNA. Okay, this is what is known as the replication. Clear? So in this session, we were talking about the Mendel's work. Okay, and then we also talked about the genes, what is a gene, and we also talked about the laws of the inheritance that was given by Mendel. Clear? In the end, we also studied about the genes. We also studied about some of the terms that are related to the gene. All right. So in the next session, we'll talk about and we'll discuss some of the objective type questions that are related to this chapter. Clear? Hope you enjoyed this session. Thank you very much for listening.